Hello!
Hi there.
Welcome to Microbiology Journal Club, where we nerd out big about all things small.
My name is Danny. In a previous life, I dropped out for my PhD in microbiology where I was infecting skin I grew with MRSA. Nowadays, I'm a fact checker for pharmaceutical advertisers and the president of Biotech Without Borders, a nonprofit dedicated aim to the public with the tools of biotech.
My name is Faz, and I have a PhD in microbiology, and I've mostly worked on bacteria, but I've also worked as a research integrity specialist, and I'm currently working as an editor for a scientific journal.
Each week we meet to talk about microbiology, and today we're doing an overview of some of the coolest microbiology papers that we've seen during the week.
And at the end of the month, we'll be selecting a paper to do a deep dive into, so make sure you message us at MicroTWJC if something has caught your fancy and you want us to take a look.
Yep. Actually, the paper that we have in the queue right now is using this weird magnet technique in order to figure out the mechanism of antiretroviral.
Yes.
Antivirals.
Magnetic tweezers. Very excited to cover that at the end of the month.
You can follow along with any of the papers that we discuss on either week in our shared Zotero library linked in the doobly-doo below.
And we want to hear from you, so please use comments or tweet at us at me at MicroTWJC. And boy, do we have a show for you today. We have some of the latest research on Omicron, where you ask the question, can it bind to cells and fuse as well as under other strains? Not only that, we also ask the question, if your lymph nodes can't form germinal centers, can you still get good antibodies against SARS-CoV-2? We'll find out.
Yeah.
But wait, there's even more. We've got a big study on how well masks work at protecting communities from SARS-CoV-2 spread. And also we look at how phages hitch a ride on bacteria to travel through the mycelial network. And a look at how minimizing the genome of bacillus can make it better at manufacturing proteins. We have the history of mRNA. And finally, a new way to genetically engineer Candida auris.
So, yeah, that's definitely a packed schedule we got for you today. And we're opening up, as always, with our SARS-CoV-2 section because that's what's on everyone's mind these days.
Yeah, that's right. So what we've got is a couple of studies looking at... So now we've got some cell culture studies coming out. This one is from Cambridge. It's a preprint stage where we haven't even got to the preprint stage because now researchers releasing their data out there to make sure that people are aware of it. So this is like so the few studies that we're going to be going through are going to be the first rubber meets the road moment. I was about to say a lot of different things with our syllables. But yeah, so here where they look at actually how the spike works. So let's see. So what they do here is
So the title of the paper is SARS-CoV-2 Omicron Spike-Mediated Immune Escape, Infectivity, and Cell-Cell Fusion.
Yes.
As you mentioned, it is a cell culture study. So they have cell cultures, but they make the Omicron spike.
Yeah. Yeah, that's right. So they make the Omicron spike and they express it on on pseudoviruses, I believe. I think it was a lentivirus one for this one. And they test it on a number of different interesting cell types. So they use lung organoids, which are stem cells that have been differentiated to form into lung cells. They also use lung cancer cells, I believe. So
two types of different lung. Oh yeah, and then a lung primary, or I'm not sure primary cell, but a cell culture.
Yeah. So
two cell cultures.
This is a common kind of way to approach cell culture because different types of cell cultures can tell you different things about and sometimes individual cell cultures might behave weirdly on their own. So what you want to do is to test as many as possible to get a general idea as to what the effect would be in a real environment. They also do some various like spike analyses that we've kind of seen somewhat before.
The neutralizing from serum neutralization.
Yeah, again, they're seeing something of what we've seen before, that booster improves serum neutralization, but Omicron is still very it's still very evasive in the antibody response. So antibodies against other strains aren't as good against Omicron, and they also take a look at different vaccines at how well they do. I mean, nothing you probably didn't hear us talk about last week.
Yeah, definitely. Figure 3 is very much last week's. The stuff coming out from last week.
Where we went.
I think in this paper, did they use AlphaFold to predict something? That I saw that they used a prediction. Let's see.
I believe so. I think that
yeah, they did. Like when they did the 3D structures here, they used like this, right? There's a machine learning, a deep learning model for protein folding, which is kind of um, a big success, I would say, in that deep learning tech,
Yeah.
allowing people to predict a bit better the shape of the things. And so like these structures that they have, this no one like has solved the structure right for um, the Omicron spike, but I guess from what I heard about Deep AlphaFold is that it is quite good, and then they also have a model right to work off of, having like the crystal structure from the original spike.
Yeah.
So, but this is just like this is that the output of those types of modelings, which then gives people a better view of like where are those mutations actually lying and how those mutations might like slightly change the conformation of the protein.
Yes, and so the thing that really attracted me to this study was the fusion studies. So what they want, so one of the important aspects of SARS-CoV-2 is when it binds to cells, it needs to fuse with the membranes. That's a really important
Yep.
factor. So Spike does this by letting itself get cleaved by some furin cleavage, or TMPRSS2 is usually what one that we go for. And so they want to test this fusing ability by using this GFP construct. So split GFP is where you take a fluorescent protein. It's a protein that fluoresces under a specific light, and they split it in half and put each half into different DNA plasmids and constructs. So individually they don't fluoresce, but when cells fuse together, they produce a signal and they turn green. It's a really neat kind of technique. So what they did was they expressed this spike on the surface of these cells, and they tried to figure out. So whenever the cells fluoresced, it's because they fused together. And what they saw was actually that Omicron fluoresced less well in these cell types,
Mm.
which is quite an interesting finding. So they found it in a number of cell types that Omicron is less good at fusing, which almost implies that it's less good at infecting lung cells, which is
Right.
kind of counterintuitive because when you start thinking of something that
Well,
spreads really fast, I mean, Delta was really characterized by the fact that it had this really good furin cleavage site that meant it could fuse cells really well.
yeah. Yeah, I think that that it seems it does seem a little bit counter, but it also might make sense from the idea that there's so many mutations, right? That like sometimes when you have that many changes, it like lowers its ability to do these other things.
Yeah,
So not being recognized by the immune system allows it to get by and like maybe persist longer inside of the circulation. But again, this is just like a hypothesis, right? Like a
hmm.
hypothesized mechanism, right? From seeing the observation. But yeah, but then having those changes might have these functional effects.
Yeah, and they checked it. So they checked in multiple cell types. I think but mostly cancer-derived cell type, not cancer, lung-derived cell types. They also did some Western blots to look at the different types of spikes. So you can sort of see that uh, there's much less S2, which is the cleaved version of spike, rather than the full-length
Right.
spike. It looked so. Yeah, this is really like really fascinating thought process here of like what could be
Yeah.
going on.
Yeah, and just to like double back, like yeah, the AlphaFold thing, like I think what they're trying to do is they're trying to say like this is like a workflow to show like some structure to function, right? Like they're saying like, look, we see like these little structural differences, right, in our predicted model, and now like it has this effect. Yeah, I don't think it's like it's previously like you'd see probably those things in a really distinct article, right? Like structure comes in its own paper because
Yeah,
there's all this methodology involved. I think the cool thing that about shoehorning, not shoehorning, but like using AlphaFold in these types of papers is that like almost anyone can do it. Like if you have the computational capacity, like why don't you like structure model the change that you think is happening? You know that there's significant caveats to like what that's actually showing you because like there's all these artifacts that we don't know, right? That are being introduced by that deep learning model. But yeah, it allows you to add like a little part to your story to try to flesh out how it connects to yeah the molecule itself.
yeah. So that's it. So next paper also paints an interesting wrinkle on that. So the title of this next paper is Human Serum from SARS-CoV-2 Vaccinated and COVID-19 Patients Shows Reduced Binding to the RBD of SARS-CoV-2 Omicron Variant. So the title doesn't really go into like why this is in history. Because this is one of the papers that has analyzed how well the Omicron actually binds to ACE2. So yeah, they produced the ribosome, sorry, not the ribosome, the receptor binding domain of SARS in insect cells as to express them, and then what they did was they analyzed it using ELISA. So they actually used two different techniques to analyze how ACE2 binds to Omicron ACE2 binds to a spike. Sorry, not Omicron ACE2. Omicron spike binds to ACE2. It's been
yeah.
it's been an early morning for me. I'm
I got what
sorry.
you were saying. Yeah. The two sticky things.
The two sticky things, the science things. Let's talk about the science stuff. Right. So again, what they find is the Omicron seems to bind less well to ACE2, which is kind of really quite interesting here. So they test it with ELISAs. They also test it with this technique called MST, microscale thermophoresis, which is somewhat like Biolayer interferometry, where essentially what you're looking at is something that gives you an output on what the basic shape of the protein is. So, so at a certain temperature, there'll be there's a fluorescence. All molecules have some degree of fluorescence, and what the idea is, if you change the temperature, that fluorescence changes, and if the binding of the molecule changes, that also changes its properties and its fluorescence. So if it's bound to something, it will change, and so from that they can try and figure out whether something is bound or not. And they do some work with that. So let's view.
Is that label free, or is that requires labels?
I think this is label free.
Okay.
I believe so.
Yeah.
Because this is a technique that I fully didn't have any idea about this morning. I've been learning about quite quickly now.
Yeah. So again, it's another line of evidence in that same hypothetical, right? Like that same hypothesis about evolution, where evolving to escape immune recognition may also involve like so many modifications that it affects some of the actual functional characteristics that we're ascribing to spike. But if we think about then looking at the data of like seeing it being a very successful virus in spreading, then obviously, right, that's not like these observations that we have of like those functional capabilities, that's not the whole story of what drives successful infectious events. But that's what makes it so curious, I think.
Yeah.
Like sort of, oh, like what is then, right, the major driver, right? And as I said, I think that one way that we could hypothesize about that is maybe it just like sits around longer in circulation, right? Like the fact that the antibodies aren't removing it from the circulation at any point, then it'll just have more chances to bind onto the thing and move in. So like, right, there's like different rates. It's all like everything in biochemistry is all about the binding rates, right? Like the K on and K off. And then, but how does that like trickle down into the expressed phenotypes that we see in disease? And this sort of discussion and hypothesis, I think, fits into like, yeah, how would you go about justifying, right, like these observations against the other ones that we see with Omicron?
Yeah. And I think that's all I'm going to be saying about this paper, really. The main thing that is interesting is that ACE2 finding. And I'm going to throw a curveball at you, Danny. Yeah. A real curveball, because I've got another study that also looks at the same question. So this one is titled Broadly Neutralizing Antibodies Overcome the SARS-CoV-2 Omicron Antigenic Shift. And
right. So there are antibodies that bind to Omicron, basically.
Yeah. So
irrespective of these changes.
Yeah, so there are like two parts of this paper. One part focuses on antibody evasion, particularly of monoclonal antibodies. So like some antibodies are broadly neutralizing, they find. Like, I think you might have heard of sotrovimab, which acts outside the ribosome binding domain. So half this podcast is me mispronouncing stuff. I'm sorry, audience members. I
mean, at least like these names aren't like some cultural, like it's not like we're mispronouncing people's names or like names of places. Like, no, these are just like molecules that are hard to say.
No, these are important technical terms. If I confuse it with something else, someone could get hurt. I mean, that's Yeah, no. So that so
So there was a monoclonal that was directed not to the receptor binding
yeah.
domain, essentially.
So yeah, they find that the monoclonals that are targeted outside the receptor binding domain tend to be better at neutralizing not Omicron. Um,
Mm-hmm.
but then there is a second part to this paper which I find which is really fascinating. Which is about the ACE2 binding. Um,
Mm-hmm.
so what they do is they do some biolayer interferometry, which is kind of like what we talked about before, where they stick uh, some protein to a surface and they check how the protein shift based on whether they bind or not. And
Yeah.
and what they found was very different from so what they found is Omicron actually binds as well as beta, but not quite as well as the alpha strain. So alpha was the
Mm.
B117 strain. So I think there were some papers talking about how that was really good at binding ACE2. Um,
Mm-hmm. Mm-hmm.
this is kind of what I would have usually expected for it. It's Omicron is slightly better at binding than other strains. So when it comes
Uh-huh.
to pay,
But I think maybe what this is, if it's BLI, then it's done with purified proteins,
yeah, that's right.
right? So like this is basically this removes. So like what could be driving the decrease is that there's also other right associations that spike could make with the surface, right?
Yeah,
And also there's co-receptor potential, right? That would drive differences. So Omicron may be deficient in those. Like I'm trying to like square away this BLI observation against the ones that we just saw.
it's quite difficult
um,
because I mean part of it is trying to figure out whether they use the same RBD for because there are different like
yes.
sub strains for Omicron as well, and
Mm-hmm.
researchers aren't always great at reporting which ones they use. Um,
Right. Like the details are there though, right? Like we just haven't done that that
yeah,
legwork for folks, like to go in and look at each of these mutations and align them and say like, oh, actually, this one might be the driving force between the different results. Yeah.
yeah, and it seems like neither of these researchers are aware of each other because I think both of them in their papers say, to our knowledge, there's no one else who's asked this question, which is always so. I expect there'll be some kind of crosstalk between them to figure out
Oh
where the
yeah.
mismatches are. But the another thing that's really
Yeah.
interesting is they also test it against ACE2 against other species. So they look at the mouse ACE2 dimer as well to see how well, and they find that Omicron binds that better than all the other strains. So that's the latter part of C. See if I can zoom in quickly. I mean, I do hate when I zoom in to a paper and it ends up smaller. Um, but that's just a thing that happens on most publisher websites. But you just have to believe me. This big red spike here is is the Omicron binding to the mouse ACE2
Yeah.
dimer, and they compared that to
Mm-hmm.
other species as well. So comparing that to the mink and others, and they found that Omicron doesn't. None of the main variants seem to bind to those as well. So it brings up this idea that Omicron could have developed in mice potentially, or they could be infective for mice. Um,
Oh, really? Interesting. Yeah,
yeah,
it does have better. It has better binding than all the rest. No, but see, like I don't think to me it's like, it's again like with the mutational profile on the receptor binding domain, right? Like, how can you have so many mutations and not change this particular property, right? That flows really well.
mm.
Those observations like mesh really well for me.
Yeah. So I mean, what we have is a good old-fashioned scientific controversy. So who's right,
Yeah.
who's wrong, or is it somewhere in between? Uh,
Yeah, yeah,
because
yeah. Well, I mean, no, I think that like I think all scientific controversies end up at the end of the day. It's like everyone was right. Like everyone's data was right. Most people's data were right. But it's like that way of talking about how it all fits together. That's like under construction.
yeah, I mean, the previous article didn't test they test the original like Wuhan variant, but they didn't test the alpha variant, and maybe
Hmm.
that could be
Hmm.
having an effect there. Than that if
Yeah, just missing that point, right? Like means that you don't have this part of the scale. So then if you imagine like zooming in, you'll see differences in a different way.
yeah.
Yeah.
And on the other hand, this study doesn't only test it using BLI, but if it tested it using ELISA, it'd come out with something different, or maybe because they both use
Right.
different testing strategies that might have their own quirks, which makes it very difficult to compare these two papers.
Yeah, yeah. I just want to say, like, I really like Figure 4 in this paper, just because it has like a nice image of like what, because I'm not sure if people are having an easy time conceptualizing. We've seen many of these papers before, like the spike protein, how it's like this big molecule, right? How mutations can get covered in different sections, and then antibodies also recognize different sections, right?
Yes.
I thought it was a nice image just to show that for people, because you can clearly see, right, like that N-terminal domain, there's a bunch of antibodies that bind really well to that, right? That region is like not in this head section of um, of the spike protein, but like the spike, the one of the receptor binding domain areas, right? Site 1 and site 2, like those are more on the exposed end of things, and there are still sites there that even the mutations in even the mutations for Omicron, like there's still antibodies that bind there, right? There's just
Yeah.
a whole bunch that don't as well. So like, just making people like keep that in mind, right? Like when we're saying like it doesn't bind as well, like there's still stuff happening in your body. It's not like your body just like this is like a stealth object and it like goes through. I mean, it is a stealth object because a subset of things can't see it, but there are still things that do see it, and like that's why we hope that that it's not going to cause more serious disease or anything like that, right? Because there are still like mechanisms in your body to remove this object, and keep it under control. It's just that a subset of those mechanisms that it evades.
Yeah. Um. So yeah, I think this paper
Yeah,
does like
yeah,
paint a
that's
good
good.
picture about like how epitopes work, and I do like how they've actually chosen to visualize that. It's visualizing
Yeah,
scientific research is a really underrated skill, uh,
yeah,
and I think that
yeah.
it's quite good when you get
Yeah.
something painted out like
And
this.
that's a really simple one, but even just and if you go up to figure 3, they have a slightly more technical one, but it's like the same idea. Like they're showing you like the footprints of these antibodies like clamping down over the span of the span of protein sequence.
Yeah, yeah.
Yeah.
Okay.
Okay, that's all.
Now the next, it's not a paper, it's a press release, and it's got very little information, but it has been
Yeah.
going through the news quite a lot.
Right.
So this comes to the eye. So this paper talks about infection in the bronchus and lung. So I'm not sure exactly. I think they say they got a primary lung model. I'm not sure exactly. So the idea is that you're taking lung cells from a living person and then
Yeah.
infecting them with SARS-CoV-2, and
Yeah,
so
I think ex vivo. So I think it's actually like pieces of tissue.
yeah,
It may actually be pieces of tissue,
yeah.
yeah.
This
Which is
happened.
like it's one step up from primary cell culture, because now we're like they're taking like the cells in their formation inside of someone's body and trying to grow that outside of the yeah.
Yeah, so the idea is like sometimes during surgery you get extra material that you're not using, and then you can
Yeah.
give it to
Hmm.
researchers. I remember in my old lab,
Yeah.
like we used to get tonsils from people because people would get their tonsils out, and there'd be that's a
Yeah.
great resource to use for testing for things in the upper respiratory tract.
Absolutely. Yeah, my roommate, we used to talk about there lots of people got like tummy tucks and things, like all the GI stuff, but
Yeah,
like, microbiome research and immune stuff, like having little pieces of the GI tract to like see how those immune cells are moving around and how they're interacting with microbiota. It's like a big data, a valuable data source.
yeah, and what they well, so the results they published of this, well, they haven't published of this. They haven't even given us a
Yeah,
preprint, so they've just given us two figures.
some
But
figures. Yeah.
we're going to try and make the most of that. Where other researchers are releasing their full papers online, even if they're in unfinished form, it's kind of a bit better than this because the great thing about a paper is you can see how each portion, how the thoughts are created. You can see the methods and the kind of how everything's put together. This one is just giving the results, and you either have to believe them, but we don't really have much context. But anyway, enough griping. What they're saying here is that Omicron is better at replicating in the bronchi than in the lungs compared to other strains. So in their lung ex vivo explants, they found that Omicron was doing less well compared to original and the Delta variant, but in the bronchus, Omicron is doing much better. So this kind of could potentially, if it turns out that Omicron produces less severe disease, this could be a reason for
Yeah,
that. If it doesn't, then this
sure.
is, but who knows?
This also reminds me of, it makes me think about like the flu, the story about flu, right? Like its receptor function and the difference that in ducks it's like a gastrointestinal thing because like they have the concentration of the receptors there, but in us it's like more of an upper respiratory thing. But this is like within a single virus, right? Like its evolutionary path is taking it through different tropisms potentially, right? Like those mutations that it's accumulating, it's not just like, yeah, it's changing the way it binds with a receptor, but in the context of tissues, that means it could be changing its tissue tropism, and that can change the way it transmits, right? Like stuff that infects higher
Yeah.
up, like maybe you cough it out easier. I don't know. Like there's all these different. It's like a very intriguing thought, right? That like if this data pans out and we can feel confident, right, in the results that they're showing, like yeah,
Yeah.
it paints an interesting picture of how the virus is changing.
Yeah, I think it also contextual. If this is true, then it also recontextualizes some of the papers we will be looking at, because those papers will mostly focusing on lung cells and lung explants, whereas if would they have done the same? Would they have behaved the same way? If they were
Yes,
looking at the
that's
bronchi,
right.
and would there be a
Right.
different protease that's cleaving that site rather than
Mm-hmm.
what the
Mm-hmm.
one we're used to? Because again,
Mm-hmm.
there's big changes to
Or
Omicron.
different co-receptors on the surface of those cells, right, is helping it get in better, right? Versus, yeah, in the lung it doesn't.
Yeah, exactly. I mean, we were seeing like contradictory stuff saying about ACE2 binding, but it could be that there's some
Yeah.
other proteins that it can bind to. As I mean, because I do remember we
Yeah.
did cover a paper on it like binding neuropilin. There could be other
Yes.
potential tropisms that Omicron might have access to that
Mm-hmm.
other strains don't, and we're way
Right.
too early
Yeah.
to
And
find
then
that
these.
out.
Exactly. Exactly.
Like this is a kind of that kind of research could take months to come out.
Mm-hmm. But also, like people, like I'm not sure if people have seen in the news, but like I think the term like endemicity is coming up more,
Mm,
right? Like the virus is becoming endemic. There's like a pathway to this virus becoming endemic. Like I think that in some ways, like a lot of the mutational stuff that we look at is trying to flesh out that story. It's like a very hard story to tell because, right, like it's not as yeah. There's so many factors that go into establishing a circulating virus population in the human right in humans, and but each of these observations can shed some small light on that larger phenomena.
yeah. Um, yeah. Let's see what else is there. I think that's all I've got to say about these few papers. Um,
Yeah.
uh, so I think that
Cool.
that moves us on to the next paper, which is still SARS-CoV-2, but not on Omicron. Um,
Yeah.
So this is SARS-CoV-2 expresses a microRNA-like small RNA able to selectively repress host genes. So this is an interesting one because I'm always interested in what the other open reading frames are doing in SARS-CoV-2. What are those other proteins that aren't spike? Um,
Mm.
and one thing, and this paper, this is one of those papers where it's quite fun because the researchers found something they didn't expect, and that became somewhat the focus of the paper. So what they were looking at
Mm.
initially was how do microRNAs in host respond to SARS-CoV-2 infection? Because all host cells have microRNAs that help to regulate proteins, and I think we've seen some papers where SARS-CoV-2 interferes with protein creation by like latching onto the ribosome.
Yep.
And the question is, well, does it do
Mm-hmm.
anything to microRNAs? What they found was in their first round of experiments, they found that it didn't, but they did find that there was an upregulation of a SARS-CoV-2 molecule in some of their cell experiments. So
Mm-hmm.
apparently what they found was that there was this microRNA-like molecule that was being churned out by SARS-CoV-2. So they follow that line of thinking down to try and look at, well, what could this possibly be doing? And it's interesting because
Yeah. So it's from one of the SARS-CoV-2 open reading
yeah,
frames.
open reading frame
Um,
7A, open reading frame 7A.
yeah. 7A. So like this is like the cool part about we I think we always touch on this when we talk about how densely packed viruses are with functionality. Right? It's like this ORF7A, like it's already making some protein that does a certain function. I think it actually is I think we've talked about before, it is an immune evading function already.
Yeah.
But even at that RNA level, like if you cut up a little piece of the RNA that comes off of that transcript, it also seems like what they're showing here is it does have seeming some immunomodulatory effect.
Yeah, apparently this has a very strong. It binds to Argonaut protein, which helps it do something towards RNA silencing, and I think they theorize it does like silence a couple of host RNA genes. But
Yeah.
their initial screen is too subtle to pick out because at that point,
Right.
infecting with the whole SARS-CoV-2 virus, you're shutting down a lot of proteins. So then they just expressed
Yeah.
it on its own to see what it was doing, and they found that it is actually doing something, but it might be doing something different because again, with it's a cell culture model, so you're limited to one cell type. But an mRNA that could be doing something different in multiple different cell types might not. So it's an interesting.
Yeah, it might not be. It might just be like the effect is real, but it's so limited in the context of what is actually happening in infection. There's something artifactual about it because it's not something that actually contributes to the lifestyle of the organism. It's hard to say that in this model, right?
Right. Yeah.
Yeah.
So, but it's an interesting idea, and it could be something interesting. I mean, yeah, trying to find out all the things that viruses do to mess with the host immune system can actually teach us something interesting about the host immune system.
It appeals to me because again, I have that framework that like everything in a virus is doing something, and so like I'm seeing this, I'm like, even if it's not contributing to real disease, it's like this is part of an evolutionary pressure that could exist, right? Like I don't think we have this paper that I kept in the potentials for a while, but I figured we may not talk about it, and I retired it. But it was about like how mutations for like antibiotic resistance change other things in the metabolism that make it more likely to do like that change the make an organism more likely to do other things. And so like listening to data like this come out like makes me think like when you remove function on things, you could uncover other functions, right? Because the effect wasn't very strong, like with all these other tools to do it, but like there's like a backup layer or something that like even if this protein became like let's say ORF7, like I haven't read deeply enough to know like what exactly the function is, but like let's say it lost its function, but this is like a compensating mechanism, like would still give like that little bit of function in certain cell types. Like that is the space in which something can evolve.
Yeah.
Yeah.
Yeah. Okay. Sorry. Oh, wait.
Oh, did the
Oh, sorry. Yeah,
the screen share
my
broke
screen
down?
share broke down, unfortunately. So hold on. Let me see if I can.
Is there anything you wanted to say about this microRNA paper?
I think that I'd basically finished up on all this microRNA paper. I just need to get the screen share back on. I don't know what Skype is playing at.
Okay. Well, if people are following along, I can tell you I have the papers at the front. So
Yeah.
the next paper we'll talk about is
Is
high-affinity neutralizing antibodies to SARS-CoV-2 can be made without T follicular helper cells.
Yeah.
So that's a pretty fascinating thought because, on our program, we've talked about T follicular helper. We've talked about antibody maturation, how that happens in the germinal center. And that's like a training program, right, for B cells. And in that training program, many cells interface with those B cells in order to sculpt the response to be something that is appropriate. But what they find in this paper is by using mouse models. So they have a mouse model, and the cool thing about mouse models is there's this huge library of promoters and ways in which that we can selectively, by interbreeding, we can create a mouse that selectively has a knockout in a specific tissue type. And so what they did here is they're specifically removing BCL6, which I think is what we talked about before in the germinal center paper. Yeah, BCL6 from CD4 T cells. So that essentially removes these follicular cells that are the ones helping the training of B cells in the germinal center.
Yeah, and this is really interesting. What they find is that they isolate antibodies from these germinal centers, and they find that some are still very active against SARS-CoV-2. That even when I remember when we talked about the BCL6 paper long ago. There's this idea that okay, well, without these germinal centers, surely it must make the immune response less good potentially because they don't have that direction from the T follicular helper cells, which are I think the kind of the dojo masters. They're the ones who are telling the B cells what antibodies to hypermutate and in response to certain antigens, and they find that actually they still produce antibodies, and
Yeah.
uh,
Strange.
yeah,
But
and
they don't say if those antibodies have the if it's clear that those antibodies have gone through like a maturation.
yeah, they do some comparisons looking at the antibody structure in terms of like whether they undergo the same amount of somatic hypermutation and whether they undergo
Oh, they do. Okay.
yeah, and they find that they actually seem to go around it seems to be fairly similar by the looks of it.
Huh.
But what they also find is that the actual diversity of so the main difference between mice with germinal centers and mice without germinal centers is that the mice without have more diversity in the antibodies. So it's not just so with the mice with germinal centers, it's like the T follicular helper cells direct them to specific antigens to react to, whereas without they react to lots and lots of different antigens. So you've got much more. So they use Shannon Diversity Index to try and characterize this, and
I see it.
so yeah, they find that what you get is a much higher diversity of antibodies against different antigens. So, which almost makes me think that this response isn't maladaptive. Maybe, but then it might.
Yeah, it definitely casts light. Like I'm remembering the paper that we talked about before. We're like, yeah, it's crazy. Like when you get infected with SARS-CoV-2, like your germinal centers get messed up, and so maybe you don't make like the right antibodies. But here they're saying they have they have data on the other side saying that like, but the diversity of antibodies is greater, and
Yeah,
yeah,
and those antibodies still react to SARS-CoV-2 in a way that is beneficial. I
right.
think high-affinity neutralizing antibodies still happen even at the germinal centers. Um, so,
Hmm.
uh, yeah, this has kind of thrown a wrench into my idea of what was going on with the germinal centers.
Yeah, yeah. And just the complexity then of understanding, like, if we see an effect, like, what does that mean for the immune response, right? Like, obviously, the immune system has like multiple layers in which it could develop the tools that it needs to defeat infection. And yeah, what they're showing here almost is, yeah, this one pathway that we think about in terms of B cell maturation, like, not the only one that might matter.
Yeah, and I think yeah, that's pretty
And I
interesting.
feel like just like having this paper in light of the way that we talked about the last one just makes me realize like we don't know a lot. We're not the best about talking about B cell like based immunity. Like just knowing the intricacies like of that cell type, it can be very deep.
Yeah, I mean, I'm just looking at this epitope map which essentially like maps like where the antibodies are hitting on the SARS-CoV-2 genome, and it's on the spike protein. And when there's BCL6, you can see these big like spikes foci where their T cells are focusing to specific epitopes, whereas below it
Yeah.
seems to be much broader and much more randomized. So it's yeah,
Yes. Yes.
this is quite an interesting paper.
Yeah.
I would like to see like
So like
yeah,
I think the idea is that like our immune system, those spikes, like that is a feature of the immune system, right? Like it's trying to help us like narrow down the place, but for different, it might not be the case that that is absolutely necessary to create the right type of antibodies to deal with something.
I'd like to hear more about this story because I think this is one of those instances where SARS-CoV-2 has just revealed something about the immune system that wasn't really as well known before. Uh, so
Or maybe just not well known to us.
yeah, not
But
well
yeah.
because specifically I haven't read many papers about this before SARS-CoV-2, so I they they've educated us, and that's important for us.
Yeah, I mean, absolutely. Interesting. Yeah, good find.
Yeah. And the next one is quite a big paper. The impact of community masking on COVID-19, a cluster randomized trial in Bangladesh.
Yes, yes, yeah. So this paper, like, it came out earlier in the month, but I was reading papers about Omicron, and I just stumbled upon this one. Like, oh, this is a great study in some ways. Very aware of, so like, it's a randomized controlled trial of masking,
Yeah.
which, well, masking-based intervention. Like, basically, the two experimental conditions that they're looking at is like one where they don't really tell anyone what to do necessarily. Maybe there's like a generalized like wear masks to keep yourself safe, but then they have an intervention where they actually tell people at community in places where people are gathering, like, you should wear a mask, like, this is why. They do like a, yeah, they do an educational intervention. And I think what I liked about this paper is it really highlights the difficulties of doing such a study, right? Like, people say, like, there's no hard, or like at the beginning of this pandemic, right? People were saying there's no hard evidence to show that masks like work, right? It's incredibly hard to grab like the highest standard of evidence there, a randomized control study, because we're working with people populations, right? Like, they describe in this paper that they just watch, that you have to watch people. Like, they had people in the community just looking around and tallying up who's wearing masks and who isn't wearing masks. And they did it in such a way that they don't want to make people feel like they're being watched, because like that, right, that could be part of the intervention, right? Like, just the feeling that people are watching you could change the way that mask behavior is going down. So what I'm very impressed about this paper is just the care that went into constructing an observation strategy and then a way to actually disentangle like this social intervention, right? Saying like, how do we educate people to wear more masks with the effect of, yeah, the effect of the study itself, I guess. They're able to disentangle this and see an effect. And what they report is, yes, this modest effect. And again, like, it's modest in the statistics that they show, but that doesn't necessarily mean that, like, if we were, like, that doesn't actually say, like, what it would mean if, like, we take the learnings from the study and apply them widely. Because the existence of an effect, I think, is very powerful to be able to show in this case. So even if that effect, I think it's like they use a ratio. They express stuff in this paper as a ratio, like basically the ratio of those in the testing, the ratio of infections and symptomatic infections in the test case as against the ratio of the against the number that are not in that test case. And so like numbers less than 1 mean that it's favoring, right, the test case. Fewer people in that case get symptomatic infection than those outside of it. Yeah, this map is like to show you how they chose the localities in which to deploy it. It's like, it's big, right? It's across the country. It's like local, like they're mixing it up so geography doesn't play a big effect, right? Like the test cases and the non-test cases are all scrambled. Yeah, it's just like the scale of the study like is very impressive.
Yeah, I've been hearing things about this study, like, bits and bobs over the past year, because I know that the one thing that I like is they had a pre-registered analysis, so they set out everything they were going to do before the study, even like how they're going to analyze the data. And a lot of studies you see sometimes they actually choose their analysis afterwards, which can be used, means that they have a lot more freedom to choose the analysis that makes their viewpoint look best or their paper look best. Whereas this one they said
Exactly.
so.
They could find the thing with the biggest effect size and say like, right, like this is how we're choosing to analyze it, and that ignores all the different statistical approaches that you could take that might show different numbers.
Yeah, it's a very sociology study thing to do, to set things out beforehand so that you don't allow yourself those sorts of like leeway. And I quite admire that. I mean, and the sociological intervention is quite interesting. They provide people with like free masks, information on the importance of mask wearing, and had mask for promoters just walk around villages to stop people who are not wearing masks and reminding them, and also partnering with local leaders and people who are just in the community as well. Because sometimes you get people run off, they throw a bunch of masks in the community and walk away, and you never hear anything from them again. And 6 months later, they go back, did you get COVID? And they go, yeah. And they go, okay, masks don't work then. Like, I still get my funding.
They really have really well documented ways in which that they did these interventions, but also, yeah, like the fact that they're separate, like they're able to separate out the localities in which they deployed those and the ones that they didn't. I think they also end up looking. They have their data set is robust enough to even look at maybe the difference between surgical masks and cloth masks.
Yeah.
And they do find that surgical masks are better at preventing spread.
Right. Yeah, this is a big paper, and this is like skating on the edge of my competence, as usual, to to understand
Yeah,
everything
no,
that's.
it's definitely harder for me to like without spending like a deep amount of time with it. Like, yeah, it is a bit hard to pierce. One of the things that I wanted to say about the pre-registration as well, what's so great about that is like, so maybe the effect sizes that they're seeing, they aren't flashy, but we can rest really assured that like they haven't biased themselves in any way to find those.
Yeah.
Yeah, and so like I think that that's like a very strong yeah, a strong element about this paper. Just
Yeah.
a really cool, a lot of work, right? For something that we don't often, especially for us, like experimental bench scientists, like this way of doing a randomized control study, like in the wild, like with people and humans, like the amount of covariates and things that like they're controlling for, and in the study, like is quite impressive.
Yeah. Uh, yeah. So essentially, what it is saying is, yeah, masks work. So I guess all those.
Nice work, yeah.
Yeah. So that
No,
means that.
maybe more importantly, it's like these interventions about masks work,
Yeah.
right?
Actually,
Like
you're right.
yeah, like it increases. They can see the effect. It increases mask wearing, and then they also see the effect. Those interventions increase or like decrease the transmission rates by this modest amount. So like yeah, I think that like connecting those three pieces is like really important. The encouragement to wear masks, right? Changing the masks that are actually worn, and then having some impact on the rates.
I mean, that's a relief for me because all those like years in like a bio in a class 2 lab where I was forced to wear a mask all for like 12 hours
Yeah.
suddenly don't feel like they were for nothing.
Right, right. It wasn't just this like thing that you did because like people said it would help, right? Like
Yeah,
there's some
like
noticeable. Eh, I mean, it's hard. Like, anyways, there's many ways to like argue for like the utility of mask wearing, right? Like don't doubt that like having material in front of your breathing holes blocking things is like it's a physical barrier, right? Like can't
right.
deny the physics of this scenario either.
Yeah, so
But
now we're
yeah,
going to end. That brings us to the end of our SARS-CoV-2 stories, and we're going to be doing some cool stories about bacteria now. Well, not bacteria. Well,
mm,
maybe about bacteria, but bacteria and viruses, and what other microbiology
yeah,
thing that has caught our eye. And this has got a bit of
right.
both.
Yeah, this one's called phage co-transport with hyphal-riding bacteria fuels bacterial invasion in a water-unsaturated microbial model system. So it's a really big connection to our cyborg soils sort of discussion, right? Where we talk about soil as this very heterogeneous environment, right? There's going to be places that are full of water, but there are going to be dry spots, air gaps, right? Like it's a big place. And somehow bacteria are able to move around in that area. And actually, in that paper, we had seen that hyphae, like the long filamentous cell extensions of fungi, end up being these bridges between environments because they're like much larger and they can have water on their surface. So then bacteria can like swim in the water along the hyphae. They're just these very important structures in the microcosmos, the micro world, right? To facilitate the bridging of the physical space. And in this paper, they then look at the phages that are on top of the bacteria and see the phages are also distributing in the environment via these hyphae.
Yeah, it's interesting. The idea here is it comes back to another paper we looked at, like which is our was it viruses using bacteria using viruses to fight each other, where the
Oh
idea
yeah.
is that the bacteria carry along these phages, and then when they go there, they spread these phages out to other bacteria who are susceptible to them and kill them off, almost using phages as weapons to colonize a new environment.
I mean, this is the guns, germs, and steel like story, but just translated into microbial ecology, right? Like, you move a virus from an environment, right, that previously didn't experience that virus. If there's susceptibility in the we'll say the native population there, then yeah, you get these like large die-off events that could be very beneficial for the invading organisms
Yeah,
in terms of opening up like resources and things.
yeah, completely. So yeah, it's hyphal-riding bacteria. I love the because again that's another new thing like where for me where bacteria can just ride on the surface of hyphae. Uh,
Yeah, no, this the words that they use to describe this phenomena, like I think are very hilarious and anthropogenic, anthropomorphic. But again, like it describes a specific way in which organisms, these at this scale, right, at the micro scale, how do organisms move around? How do things get mixed, right? Because in marine environments and in sort of like fully submerged environments, that problem is generally thought of as like, yeah, currents and like flow and like everything's contiguous, right, in the
mm,
water, and like, right, you can have force propagate. But in soil, there's tons of soil environments, like those small air gaps provide these are really big barriers in some ways. And like you can ask like, how do things get mixed, and how did like the little isolated, like within a centimeter cubed, right, of soil, there could be all these distinct communities
yeah,
right that are going on. And what this paper is providing evidence for is that like hyphal growth in that community can bridge those spaces physically that then let the biological components intermix, which then causes changes to the overall population structure, because maybe the only thing that was stopping an outbreak of the particular bacteriophage in like the upper millimeter square of this centimeter square, right, was its isolation. And a little hyphae bridge will then allow that phage to access that population of bacteria and wipe it out, thereby opening new resources for other bacteria to grow in.
yeah, and it's good for the phage as
Yeah.
well because the phage just gets more bacteria to infect and propagate. And yeah, it's another like paper looking at the community and how important fungi are to creating these environments. Um,
Yes, yes, yeah. How big of a factor they are in so many things, because at that scale, right, like these large hyphal structures provide the scaffolding of activity, right? That this sort of like complex, chaotic life is like built off of.
Yeah, I mean, we often forget because when you grow bacteria in petri dishes or in like lab cultures, they tend to be quite homogeneous. But in real life, they tend to grow in very heterogeneous communities in the soil, and
Yes,
you
yeah.
often lose that perspective. When I guess they did it here quite simply by carving a little gap in their agars and let the fungi bridge it.
Yeah. Yeah, yeah. I love like that. Yeah, just that small thing, yeah, gives us this story that helps us understand the complexity of what's going on in soil, basically.
Yeah, are you guys?
Okay, that's all I want to say about that. Um,
So now, next paper is redirected stress responses in a genome-minimized midiBacillus strain with enhanced capacity for protein secretion.
hmm So I'm not sure if people are familiar with Bacillus subtilis, but that's a gram-positive rod spore former, very basic workhorse, I think, in gram-positive genomics. And there's a significant project from bacterial geneticists where they ask the question, what's the smallest genome possible? and and it's sort of an intellectual exercise to understand like what is an essential gene in a genome versus an inessential gene, and then trying to like remove genes to then create a minimal life form. And by doing so, it increases our understanding of sort of the interdependency of genes in a network that goes into making life. So this midiBacillus strain is a product of this scientific like question, right, to explore genome structure and sort of genome regulation by creating a minimal Bacillus subtilis strain. So there's actually a mini bacillus, I think, as well. This mini bacillus is kind of not not the most extreme version of removing every gene that they deem inessential, but rather some midpoint of that.
Mm.
And they are taking this midiBacillus strain with the idea that, well, hey, they removed a lot of stuff, so maybe this bacillus is going to be better at making stuff that we want it to make, because it's just has less of that, those sort of like noisy circuits, right? Noisy genetic circuits in it. And so they do that experiment where they induce protein expression both in wild type Bacillus subtilis and this midiBacillus, and they find, oh wow, like it really does make protein better. And they try to then track that down to gene regulation networks. So like, yeah, this figure that we're looking at, these complicated Voronoi graphs, are essentially like every gene takes up like a little bit of the area of these graphs. They're grouped by their function, so that like spatially functions kind of group together, and then they get colored based on either the induced, like whether they're being turned on, right, when the protein's being induced versus the protein not being induced. And I personally can't read these at all, like in any meaningful way. Do you
So
have
yeah,
any insight?
my insight is look, there's more brown patches in the midiBacillus in this place, which kind of indicates that there's more protein synthesis going on because they labeled this Voronoi square with protein synthesis. And coupling with stress,
Right.
there seems to be a lot more proteins out there being coupling with stress. But yeah, this seems like a magic eye portrait to me somewhat. So I can understand, like I can see a sailboat, I think. But
Yeah,
yeah.
they're hard to read in that sense. Like, yeah, like I guess there are more only induced, like partially induced genes in the midiBacillus like thing, but I think the meaning really comes out when I was reading the text of this paper, or
Mm.
skimming the text of this paper. The meaning comes out more like with the actual annotations to the genes that are being turned on. They sort of identify that, yeah, there are these stress response, like the stress response is very different in the induction in the midiBacillus, and in a way that seems favorable to the production of more protein.
Yes.
So it's like, figure 5 is actually really interesting. It's nothing about like the activity of transcription in these minimized genome strains that is making them better producers. So like typically in synthetic, if people are like trained in like central dogma, right? Like DNA makes RNA makes protein, right? So the rate of making RNA, the rate of making protein from the precursor, that, right, from first principles, you would say that is what we need to control to get more proteins out. but what they're showing here is that, well, actually the midi and the midiBacillus, they're actually not very transcriptionally active. They're not like making tons of transcripts, right? And that's not the flux that's being controlled. Something else is controlling getting all the protein out there,
I mean,
and they tie that to like stress responses.
Yeah, this actually reminds me of like a paper I think I glanced at recently called, I think, Cells Are Just Bags of RNA, where RNA is just doing so much other things that we don't really appreciate other than creating proteins. And so
Yeah.
so actually this kind of the kind of dislocation between like protein production and RNA isn't necessarily as disastrous to protein production as we'd expect. So it's
Exactly. Yeah, there's like it's not so simple as like I make more of this transcript, I'm gonna get more of this stuff. I mean, like in vitro, I think we see that relationship, but like that's not the governing relationship inside of a cell, or not the be all governing relationship. There's all sorts of things. So yeah, I think this is maybe figure 6 is the real money making shot, right? This is where we see, oh yeah, there's a lot more of this heterologous protein. It's a virulence factor. I think they're making ISAA. midiBacillus just makes a whole bunch more.
yeah.
It's able to.
It's a virulence factor that's produced by Staphylococcus aureus that the parental strain can't make, weirdly. So
Yes.
some reason, some part of the irregulation that would prevent it from being making has been removed. And I think this might be like a vaccine candidate potentially. I'm not quite sure, but like the idea of like targeting a virulence factor means that you're targeting like the bacteria in a phase when it's actually virulent and benefiting the commensal forms. Yeah.
Yep. Yeah, you do get some production, right? Like if you go back to that figure, like the parental strain in the cell fraction in the
Yeah.
induced, right? So you get stuff, but it's not well secreted. It's like stuck in the cell, right? Like They don't like making this protein, and that could be, like that's going to be true for so many things, especially if they have enzymatic function and stuff like that,
Right.
right? Like there's so many things that can mess up the inside of a living organism if you're trying to make them at like industrial scale manufacture. And so again, like it's a really attractive thought and very cool the way in which a very basic like scientific research object, right? The midiBacillus, right? Like there's no reason to have made that technologically speaking, right? It's about uncovering like what the minimum genes are, but here's a great function of like taking that research object and saying it's going to have industrial impact, understanding these things.
Yeah. I don't think I've got much more to say about that. It's got an exciting
Yeah,
because yeah,
I think that that's what's to say. I mean, they go in and they talk about all the different genes they think are being turned on and turned off, but I don't have the the understanding to be able to discuss that really.
yeah. So let's move on to the next paper, which is evolutionary processes driving the rise and fall of Staphylococcus aureus ST239, a dominant hybrid pathogen.
Yes. So I wanted to bring this one up because I mean I've worked on Staph aureus and this was a story that I was very interested in always, like the evolution of Staph aureus. It's a pathogen that at the beginning, MRSA, methicillin-resistant Staph aureus, it was found associated with hospitals, and people were like, "Yeah, this is like evolution that happens in hospitals, right? Where hospitals, because we use antibiotics, some bacteria have filled the niche where they're like hyper virulent essentially and living in hospitals because there's all this opportunity if they can just get past the antibiotics. But then people found like community spread versions of these, and it's just been like it's just an incredible and I'm not also I've kind of forgotten a lot of it, but like it's an incredible story of like the spread of antibiotic resistance and how like acquiring a cassette like that gives you access to different environments. So in this paper, they're sort of they're fleshing out that story even more by looking at many different sequences, specifically focusing on this ST239 variant that really exploded on the scene at some point, right? There was a point in the history of staph where this was the MRSA strain that was moving around, and it was known that it's from a recombination event, and they kind of talk about it a little bit more here, where phage mediated recombination moved in a whole cassette of things into the strain, moved like a huge amount right, of chromosomal information, and that gave the hybrid strain a more favorable transmission or like a resistance profile that let it be so successful. Um, let's see, what else do they want to say here? And then what they show in this paper, yeah. So the next couple ones are like just illustrating that recombination event,
Yeah.
right? How do they know that this recombination event actually happened? Based on looking at it in relation to other genomes, and then they look at they say, oh, this recombination makes it more competitive. It's able to, that's right, assigning the function to this huge influx of genes. But then they also look at then the drift
Yeah.
from that recombination event, right? Because like that, presumably that recombination event happened once, but then other mutations started accumulating on it. And the speed at which the mutations are accumulating on that gene, I guess, is like also telling something about the story of how it's also a very energetically expensive like set of genes to all of a sudden have in your genome, and so it's impacted them. It's impacted the strain in other ways where, although it might be successful in resistance and stuff, it also has fitness costs, and now we don't see, like, this is a story of the past, right? This is not the dominant strain anymore. There are other dominant strains of Staph aureus, and this is sort of addressing that part of the story as well.
Yeah, I like this because I mean I feel like this sort of thing I'm getting a much better perspective of thinking about SARS-CoV-2 variants. That's taught me a lot about how and what
Uh-huh.
we're seeing here is somewhat like that with, I think, sequence type is what ST stands for. I'm not sure. Uh, but
Yes.
but yeah, looking and seeing how different sequence like evolve. But it's like over because I think this was first spotted in maybe 1959 or somewhat like
Hmm.
that, and yeah, and it's taken like 30 years for it to become dominant, and then it's taken a decrease in the past few years.
Yes. Yes. Yeah. It's like over this time. And I think the other thing is important. Yeah. So ST sequence type, it's because they didn't do these through whole genome sequencing back in the day,
Mm.
right? They did them through like collections of like loci that they knew, right? Like so like a DNA fingerprint style, right?
Yeah.
Or targeted PCR of specific genes and alignment of those specific genes gave rise to like a sequence type system. So it's not a full like whole genome thing. And I guess that's also why this paper is very interesting, is because now we just sequence everything, right? And make those alignments. We can tell that story with more resolution. And again, like we can also tell the story now of like the mutations that have accumulated over time, right, in these strains, and like that drive the differences. So that's yeah, that's what this paper is about.
Yeah. And that brings us to the last
Yeah.
paper. Forward
Oh
and
wait. I just want
Oh, okay.
wait,
Sorry.
one last thing.
Doesn't bring us to the last
Just
paper.
no,
Rewind.
and the takeaway there is just right like, and this is like I feel very passionate about this because like I love bacteria. Like I just think they're so cool, and pathogenesis is like such a interesting complex process, right? Add that to evolution and you get these like very long running stories in our society, in our world, right, of bacteria trying to make the best of their environments and understanding that relationship, especially as it relates to the environment of the human body, I think is like a very important element of health and surveillance, right? That maybe gets undercut sometimes by thinking about like therapeutic applications, and like we want to make something to solve like an existing disease problem, but understanding some of these dynamic elements of how like the ecosystem changes and that drives disease in certain ways, just to me is like an important piece of like maybe a more holistic view of what it means to take care of human health. Right? So surveillance is part of human health is how I want to like have people take away from these types of stories.
Right.
Yeah.
Now that brings us to the to the next paper. Forward and reverse
Yeah.
genetic dissection of morphogenesis identifies filament competent Candida auris strains.
Hmm. Yeah, so Candida auris, I'm not sure if people have heard, this is like a new scary pathogen that's emerging inside of hospitals. Very drug resistant is the fear of this particular pathogenic fungus. It's a yeast,
Mm.
essentially, right? Candida, we know, is a very yeast
Yeah.
infection is caused by a Candida strain. And Candida, or Candida species.
Yes.
Candida auris, yeah, a very new emerging pathogen, especially inside of hospital settings. Another reason to examine hospital infrastructure and how things are done there. And what is cool here is that they're showing us basically a toolkit inside of genetics. So forward genetics is this process of breaking things in the genome and then figuring out what phenotypes come out, thereby learning stuff about what you broke. And reverse genetics is taking something that has an interesting phenotype, then breaking the thing that you think, then making the change in it, right, to track that or to track down the point in the genome that caused that thing. And so they use both those tools here. The forward method they're using is this transposon library using
Yeah,
agrobacterium.
agrobacterium to
So,
transform fungi. I never thought didn't know you
uh-huh.
could do that, but apparently it's yeah, you can
Me neither. I guess it's the way to do
yeah.
it.
Because usually agrobacterium
um,
is associated with plants, so I think it was
for
yeah, it
plants,
lives in the
yes.
roots of plants as like a pathogen and it manipulates the plant's genomes to create these little kind of compartments for it inside those plants. And researchers managed to take
That's
that and
right.
use it to genetically manipulate plants, and now apparently it works in funguses as well, which is really interesting.
That's right. And so the payload that normally these bacteria would deliver is like, some sort of thing that changes the development of plant nodules. But here they replace that payload with a transposon style payload. So like that's a jumping gene, you might have heard. And so it jumps out of what was in and it jumps randomly into the genome. And so basically that process breaks things. And so if you do that across like a whole bunch of different bacteria, or a whole bunch of your target bacteria, the Candida in this case, they create a library where each Candida has a lesion somewhere in its genome. And then they can test each one of those bacteria, they can, or microbes, yeast in this case, and you get something like B, right? Where you like have plated out all these different bacteria. The theory is from every single bacteria grows a colony. When that colony looks kind of wonky, you're like, well, something happened in the genome there. Let's pick it out and find out what we broke.
Yeah, and that's what they did here. And I think this is because, again, when you have a new strain of any life form, it takes a while to actually figure out how to do genetics on it, because sometimes they have different little. So actually, like, part of this is that model of how they're doing it, and then next part is a proof of concept, doing something interesting with it.
Totally. Yeah. So they found like, oh, it looks kind of funny, and they see a difference between like filamentous versions of the organism and ones that have that broken in some ways, where they get stuck together, I guess, more frequently.
So, yeah, they got this picture of these normal like cell colony types, and then they have these various mutants, and looking at like the microcolonies. So like when you're at the scale of tens of cells, and yeah, that's quite an interesting look at like how the structure. And then because they have this tDNA like codon, they know the structure. They can figure out where it is and what it's affecting.
That's right. Yeah, so they've been able to now, like on a really large scale, scan over the genome, find out several genes contribute to a similar looking weird ass colony morphology, and that really is the toehold that people need to start doing in-depth, right, mechanistic analysis of what's going on here.
Yeah, and so they also do some Cas9.
Yeah, they verify. So like a big part of this is like once you've done it this way and you found things through the screen, then you go back and you like make specific deletions in those areas so that you haven't. It's basically it's a verification, right? That like what you're doing isn't just some artifact of that experimental system. You can make the same change. You can make a cleaner change, right, in the genome using CRISPR. Just like remove it scarless style. And still see some sort of weird clumping phenotype.
Yeah.
Yeah. Well, this is them using that, using it. Yeah.
Yeah. Okay. And I think that. I mean, do you have anything else we want to say about this? Or
no, I guess that's all. Like that's what they did.
Yeah.
They went from breaking everything randomly, then they verified their responses. It's a great new tool set because Candida auris is like this emerging pathogen. It's important that we have these tools because it allows us to understand like what those specific mechanisms of pathogenesis are that then hopefully lead to a better understanding of the therapeutics we can use and maybe even the themes of pathogenesis in general, which just help us understand all pathogens more broadly.
Yeah.
And yeah, that's all I want to say.
Yeah, no, I think has some good papers on like toolkits to use for bioengineering with was it the backlist minimized the backlist, the transposing constructs. We've had some. Yeah, this has been quite good. We've had some highs, had some lows during the show, but unfortunately, all great things must come to an end. So.
Yeah, so join us next week for more cool microbiology news.
We want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's possible we didn't get everything right. So science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the comments. And also, I'd like to thank all the people who commented as well during this. It's been really informative and really helpful.
Yes, yeah, it's great to have more commenters. We definitely enjoy that dialogue, and we're hoping to have that dialogue with more and more people. So tell your friends.
Like, subscribe, and the third thing that I can never remember.
Yeah, you can reach out to us over the hashtag #MicroTWJC or add us on Twitter. Peer review, we both believe that peer review is this process really of talking about papers and so that we can all participate in it, and we hope that you've enjoyed hearing us ramble on about microbiology today.
It's been a pleasure chatting with you, Danny.
Same here, Fuzz.
Tune in next week for more microbiology content. See you later. Bye.